And I'm sorry we went over a little bit for our next speaker, who is Professor Jacqueline Linnes. She's an assistant professor in biomedical engineering, and I think the reason I'm most excited to hear her talk is I think she shares really that aspiration that a lot of us in the point of care field have, to test the world and, provide more actionable results and information to people. And so I welcome her talk now.
Thank you. It's really an honor to be here. I really appreciate this opportunity, and I'm going to tell you about a couple of the projects in my lab. We do, I'm going to go back a second, quite a bit of work focusing on paper-based devices and on microfluidics for point-of-care technologies that can be used anywhere in the world, mostly around infectious diseases, but also we have some projects in non-invasive sample collection from exhaled breath, so that we could monitor glucose or maybe COVID-19. And another project around wearable devices, where we're working with people who are interested in opioids overdose detection. And so it's actually been really an honor to work with all three of our other speakers today, Frederic and Shreyas and Steve, on various projects in the lab. And I'm going to talk only about the two focused very specifically on diagnostics. The first being the particle diffusometry that Dr. Worley had mentioned, where we are focused on leveraging this for detecting pathogens in this case, cholera detection in a water sample, so that you could take this device out to, let's see, we've brought it to Haiti, to Bangladesh, to Kenya, anywhere that there could be a potential epidemic of cholera, where you could test the water samples and detect whether it's there based on capturing the pathogens and then lysing the bacteria in this case, amplifying the DNA, and then detecting the long chains of DNA that are made using particle diffusometry, where if the sample is there, then it gets very viscous and you can see a slowdown. And so a lot has gone into the background to that, but for the user, you simply collect a sample, heat it on a chip, and then we're actually getting rid of the add the particles separate that's incorporated into the chip, and then image that on the phone using that particle diffusometry that Steve had mentioned. And like I said, typically you would capture and amplify the DNA, and that would be monitored using fluorescence. But because we make these really long strands, we can use particle diffusometry here. And because the videos don't work, they look very similar. But as Steve said, low diffusivity here would, I'm going to see if I can get the little pointer to work too. Yeah. Low diffusivity would allow us, the particles would move very slowly, and high diffusivity, they move much more quickly. And all of this works on the smartphone. My lab has collaborated very closely with Steve on the hardware side of this, where we have made our own little microscope, which is a laser pointer, now an actual slightly higher powered laser, and some theater film and a tiny little grain of sand sized ball that sits in there to magnify the particles, so that you can see these 400 nanometer particles on the phone itself. And then over here's a little heater. In this older prototype, that's a paperclip, but it's gotten a little fancier now, but we put it together to make it work initially, and we've started a company, Omniviz, that's actually translating this technology and making it robust so that it can work out into the field, and my lab's focused mainly on this test chip side of things and the biologic portion, as we're working to get the assays robust and specific and sensitive so that we don't have any detection of non-pathogen samples, because there is Vibrio species that are, Vibrio cholera is one of them, that are non-pathogenic in the environment, and they're not going to cause a horrible diarrheal disease. And we don't want to accidentally detect those. So we focus on that toxin gene itself. And let's see, we're stuck. There we go. And it has been a large collaboration to get this off the ground. I've worked on the nucleic acid amplification assay, Steve, the microfluidics, Dr. Tamara Kinza-Ursum, the actual nanoparticle conjugation, and she and Steve developed the particle diffusometry, along with, on the other side of the circle, Dr. Catherine Clayton, who's the CEO of OmniVis, and is really passionate about bringing things out of the lab. This project has really grown, and so now we've got collaborators not just on, can we get a technology piece to work, but is it going to be usable, and what do people actually need to use it for? And Dr. Ronald Tanwi, who's at Moi University in Kenya, and they're actually using this. And I realize that's not the right picture there, but it is a smartphone device. And so, as Frederic had mentioned, it really is the students involved that do most of the actual work in the lab, but we all get to take the credit. But without the students doing the work, it wouldn't really never be able to get done. And so we need to keep them in mind as well. Another project that we're working on is this paper-based device. And this, our goal is to combine the simplicity of a lot of those paper tests that you hear about for example, COVID-19, or essentially these are pregnancy test style assays. They're an immunoassay. And we want to combine those with the accuracy of lab-based nucleic acid diagnostics, because those are the gold standard in many cases. So our goal is to amplify to capture the bacteria or the virus, amplify the DNA and the RNA, and then read out just like these protein assays, where you have one line for a negative sample and 2 lines for a positive sample, to show that you can see all of these complicated things that the user doesn't have to know anything about. So here is a concept of a sample separation membrane. We started with HIV. So in this case, we would separate blood from the virus and the plasma, and then move that downstream to lyse and amplify, reverse transcribe the RNA to DNA, amplify that, and then move that again further downstream to the detection zone. Here we are controlling this with wax valves, because wax is convenient and melts on demand, and then the liquid can push past that valve in order to move to the next zone. And come on, little buddy. This is a smartphone-powered prototype of that device, and I've got it sitting here. So it's about the size of a digital pregnancy test, and we were able to put those valves in different zones, and the reaction zones and the separation zones. It happens to be powered by a smartphone because we need the battery off of it, but you can imagine, and I would like to take this forward so that we're actually leveraging the power of the smartphone, similar to the OmniVis project, where you have that computational aspect and you have the connectivity aspect, which are really critical in moving forward with not just detecting something, but then connecting people up to care for the next steps of what do you need to do next. And so for the user, as I mentioned, it's very simple. You add a sample of blood and some wash buffer behind it. You fold it up. As soon as you plug it in, it starts to heat. And then it does that heating and walking through each of the steps, depending on the pathogen that we're looking at. That takes 30 to 90 minutes. And then you can read out positive, 2 lines yes, 1 line no. As I mentioned, we showed this for HIV detection in blood. And one of the things that we have been focusing on my lab is the usability. And so on the right side, we quantified the line intensity. And we also looked at what line intensity can somebody see if they're in the sunlight, if they're in the shade, if they're indoors, because you don't want to have This, again, resulted from a lot of collaboration. In order to be able to do the heating on chip, we worked with Dr. Leah Stancia's lab that has really thin film heaters that use low power printed silver ink. We've been starting up reagent storage in collaboration with the LIO Hub here at Purdue, and Alina Alexenko and Liz Top are leading that. We took this device to Kenya, and Professor Eddie O'Darry has been testing that out. And again, Natalia Rodriguez on our user-centered design side of things. We've got a new project moving forward. And then what's really critical is not just can we make this device in the lab one at a time or a few at a time, but can we make it a scalable device? And so Purdue's got a reel-to-reel manufacturing center, and we've initially started working with Dr. George Chu on how can we put in the proper controls in the process of designing this and manufacturing it so that we have the reliability that we need to produce hundreds of thousands or millions of these devices.
Thank you. So I guess we did pretty good. We're 1 minute over all. So I'm gonna forego any further questions on processes and stuff if you don't mind, and allow Jo Beth to take over at this point.
Great, thank you so much to all of our panelists today. And I think we had a great first virtual frameworks. I want to again thank our sponsors, Indiana Biosciences Research Institute, for helping us put this on, and Dr. Pooja for putting this great group together. All great information and exciting work being done here in the Indiana Life Sciences community. Also want to thank our sponsor, Indy Science Connect, for helping us get the word out. We will be having more of these moving forward and throughout the years. So you can go to our website, biocrossroads.com, to sign up for a newsletter if you aren't already receiving notifications, or you could follow us on Twitter if you'd like to know about more of these virtual frameworks. And with that, we'd like to thank everybody for coming and have a wonderful day. Thank you.